So hi, everybody. Welcome to the first day of our community workshop. This workshop is meant to facilitate crosstalk and network building across working group. So during this workshop, the three working group each day will present about their work and will gather public feedback on specific questions related to data sharing and archiving in agriculture. So we encourage everybody to contribute with their feedback and experience. Everybody that works with data, that curate data, and that as a librarian, a data curator, a data scientist is very important in our mission to make guidelines for fair data in agriculture. Before starting, I want to remind everybody that AgBioData is a code of conduct. So we promote a safe and productive environment where open dialogue is allowed and that where everybody feels comfortable in sharing their ideas. We promote equal opportunities and treatment for all participants, and this environment is free of harassment and discrimination. So if you want to hear more about, if you want to know more about the code of conduct, you find it on our website. If you are experiencing or you are noticing harassment or misconduct and you want to report it, please send an email to agbioData@gmail.com or to Susan McCarthy at askmccarthy.agbiodata@gmail.com. Again, these contacts are on our website. And so with this in mind, I want to provide some suggestions for today's discussion. We are encouraging everybody to share their opinion, and as we are also respecting other people's opinion regardless of their position. So we listen to them and we don't interrupt and we show empathy. I also encourage the Guardian, the host during the breakout room to follow this principle and to make the discussion inclusive and respectful. And with this, this is the general agenda that we will have each of these 3 days. So each day will start with the 3 working groups presenting their work. The presentation will be of 10 minutes, followed by 5 minutes of Q&A session each. Then there will be a short break of 10 minutes and then a breakout room section of 45 minutes. Each breakout room will have an host, which is a person of the working group that will lead the discussion and will try to promote an inclusive environment. This person will also report at the end of the discussion about what has been discussed. There is a scribe, which is a person that will be responsible of taking notes of the discussion. And then there is the Guardian. The Guardian is a steering committee member, that will supervise that everything is going fine. This person is also a co-host, so will help me with the logistic and also can also actively participate in the discussion. And then everything will end with short lightning talks, 5 minutes each, where the working group will present what has been discussed during the breakout room session. And with this, I want to introduce today working groups. We will have the Genotype-to-phenotype with Irene Cobo Simon presenting about their work. Then we will have Rice presenting for the Standard for Genetic Variation Working Group. And finally, Doreen Nestle presenting about the Recruiting Working Group. And so I want to remind everybody that if you have any question, please feel free to write them in the chat, and I will be happy to ask the speakers to answer them when they will finish their presentation. And with this, I want to invite Irene to share the screen and start her presentation.
Thank you.
Becca.
Okay. Okay, it is, can you see my screen? Yeah. Okay, thank you so much. Hi everyone. Thank you. First of all, thank you very much for being here today. My name is Irene Cobo. I'm a postdoc in the Department of Ecology and Evolutionary Biology at the University of Connecticut. And I'm also the chair of the Phenotype Genotype Working Group. And today I'm going to present the most recent updates of our working group. First of all, I'm going to start with a quick description of our Genotype-Phenotype working group, like in particular the main scope and objectives of our working group. Then I'm going to explain a little bit our most recent updates as a working group. And finally, I'm going to answer the question whether we are looking for more members or not in our working group. So first of all, regarding the scope and objectives, our main objective is basically to improve the data collection and data sharing in order to help the identification of genotype-phenotype associations. And to this end, we think that it is essential to better define the data and also the metadata for this phenotype and genotype studies using standardized terms, for instance, ontology. So the metadata is basically information about the data about the data. So for instance, how the data was collected, the study design, for instance, all this information about the data. And this is necessary in order to ensure the so-called fairness of the data, that is, its findability, accessibility, interoperability, and reusability. These genotype-phenotype associations are, sorry? Okay. These genotype-phenotype associations are important in our, nowadays, are relevant and timely nowadays because several current economic, environmental, and social needs can be addressed by these genotype-phenotype associations. So for instance, these associations can help identification of genes controlling traits, providing resilience or tolerance to several biotic and abiotic stresses. And this is especially important in the context of a changing climate. And these genes can be used in conservation strategies or management strategies. So this is important from an environmental perspective, but not only from an environmental perspective, because several model species that are used in these genotype-phenotype associations has a economical or, and as a consequence, social importance. So for instance, in the case of crops, candidate gene identification can help enhance the plant health and the productivity of these crops, which is also important from a social and an economical perspective. In order to accomplish our objectives, we need to identify, on the one hand, what is already done in this topic in order to avoid repetition, and also what it needs to be done to make the most of these genotype-phenotype associations. And this is especially important in the current multi-omics era, which is a challenge for data storage, integration, and analysis. These omics techniques that are represented in this graph that are phenomics, metabolomics, proteomics, transcriptomics, epigenomics, and genomics, all of them produce a huge amount of data and a very complex data. So this is a challenge for data storage and also for data analysis. But another challenge is this data integration. So it is very useful to try to integrate the results of all these omic techniques by means of the so-called system biology approach in order to help, because it helps elucidate like the biological processes, the metabolic routes that are behind the traits of interest. But that supposes a challenge for data integration given the complexity of the data and the huge amount of the data. And another challenge for data integration is that many of these techniques, these omic techniques, coexist with data coming from older techniques. For instance, in the case of phenomics, there are also phenotypic data collected using more, we can say, traditional techniques, which is another challenge for data integration. So in order to better define our objectives as a working group, because all this field is huge, it's very complex, we come up with the idea of writing a review paper that we have entitled it Linking Genotype and Phenotype in Multi-Omics Era. And on the one hand, this review paper, as I have mentioned before, we help us better define our objectives as a working group by on the one hand, identifying the challenges and the limitation of linking genotype and phenotype data in this multi-omics era. And on the other hand, to help us figure out how to ensure the fairness of this data. As I have mentioned before, one way to do so is by improving the metadata collection using these ontologies and standards, standardized terms. Also by making this data open access, and there are another options. But at the same time, this review paper not only help us to better define our objectives as a working group, but also we think that will be a very useful resource for all the scientific community. And to do so, we need your feedback. So we are going to use our breakout room session to get your feedback about the current challenges and needs that you are facing, because many of you for sure will be are dealing day by day, like in a daily basis with this kind of data. So that will be very useful for us in order to write the review paper, to know what are the challenges and the needs that you are facing day by day in these genotype-phenotype associations, not only in producing this data, but also in using this type of data in this multi-omics era. In addition, we want to discuss another, like we can say, difficult to define aspects of these genotypic-phenotypic associations. So for instance, how to define a phenotype, which is a very vague term. So if we can consider, for instance, gene expression a phenotype, but this is an example, but we can discuss another aspects that you want to discuss. And finally, we want your input about the structure or the scope of this review paper. And regarding the last question that about if whether we are looking for more members or not, the answer is yes. We think that we could benefit more from someone with an animal-related background, but everybody interested in joining our working group is very welcome. So if you want to join our working group, you can write me. I'm the chair of the working group. This is my email. And also you can write the co-chair of the working group, who is Ines Rebollo, and this is email. Feel free to write both of us if you want to join us. You are very, very welcome. And finally, thank you very much for your attention.
Thanks, Irene, for your presentation. If you guys have any question, you are free to ask to the speaker now. So,
Hi, this is Bob Cunningham. I'm curious what your experiences have been so far in the working group with ontologies, and how people have used them and what their experience with them has been.
In our working group, you mean? So far, we are still defining our objectives, and we are like writing this review paper in order to better define them. But yeah, I'm personally working in a project that precisely is trying to help improve these ontologies and standards. Mostly, we are working with both genotypic and phenotypic data. So we are using, for instance, MIAPE. I don't know if you know the MIAPE, which is like it is for phenotypic data. Is a minimal standardized terms for phenotypic data, and we are also trying to standardize the metadata for genotypic data, which has been a little bit more challenges given the different types of data that we have in the project. But yeah, the project that I am working with is called Cartograplan. It's an application that integrates all this type of data, and also environmental data, which is even a little bit more challenging for us. So yeah, I have experience with that, but as a working group, we still are trying to figuring out what is our scope and objectives.
Okay, thanks.
Thank you.
Irene, there is another question. What group systems are you addressing, or are these more of a guideline for all the systems?
What groups I am personally addressing, you mean? I'm working currently my project I'm trying to integrate maize, but this is the only crop, proper crop that I am working with. Our project is initially started with trees, so the majority of our species are trees. But
I think question is for this review paper. That so I think we are here the guideline that Irene is providing for all the systems.
Mm-hmm. And so what is your question? Sorry.
So question was from Raj, that what crops and systems we are going to address in the review paper.
Oh, But that's a good question. Actually, we can discuss it in the breakout groups.
Yeah.
Yeah, that would be a very, very good topic. Thank you.
Well, if there are not other questions, I have a question for you, Irene. So the Genotype-to-phenotype working group is a very broad, like there are many things. Do you see any connection with other working groups that we currently have, and do you are willing to collaborate with any of them specifically? Like, there are overlapping topics with some of them.
Oh yeah, for sure. Yeah, this is another concern that we have. For instance, the standards for genetic variation, that will be a good group to collaborate with, because there is a kind of overlap. This is another reason why we need first to establish what are our objectives as a working group, because there are a kind of overlap between groups and we want to avoid this repetition, and also just collaborate with them in order to make the most, like just exchange ideas and everything. So that will be, for instance, this group will be like very, very, very useful to collaborate with.
Okay, yeah, so I think this is the last question. Monica is suggesting like the AgBioData. Is AgBioData working on similar problems? And if so, have you reached out to them? You know, what is AgBioData?
Working on similar problems. Well, actually, I have a in my project, in which I'm working as a postdoc. I have a AG2PI seed grant, and I'm addressing the same issue. So yeah, for sure, they are working in similar problem. So yeah.
Well, thank you, Irene. That's very nice. So I invite everybody that is interested in the Genotype-to-phenotype working group to join them during the breakout session. And so I want to invite now Rise for the standard genetic variation to share the screen and start the presentation. Rise, are you there?
Yes, I'm working on the share this screen.
Okay.
Just wait a moment. Okay.
We cannot see your screen yet.
Hang on.
Otherwise, if Marcella has the presentation.
Is it?
Okay, yes, great, thank you. Oh, I want just also to say that the standard for genetic variation has a poll question at the end, so stay there.
Yes, so we will have the poll question at the end. So welcome to everyone to participate. So good day, everyone. My name is Taoho Chang, also known as Rice. I'm a co-chair for the Genetic Variation Working Group. And today I would like to present about our output at the standards for genetic variations. So to begin with, we would like to heads up to everyone that we are certainly looking for more members to join our working group. And later on, I will have a brief introduction about our standards for genetic variation, about who we are and what is our scope and objective. And for the last part, we will focus on our progress update and the challenges at the moment we have got. And the end, we will have a live poll. So welcome everyone to join us to have some ideas to contribute. So we currently have members from US and Germany, UK, and me from Taiwan. And so we are looking for more members to join us to have ideas for the standards on the genetic variation. Sorry.
So listen, the key takeaways here are basically understand the requirements for this. Because this is project management, I tend to say that if you miss handing something in on time,
I can't hear the question very clearly.
Okay, Rice, you can go on if you want with the presentation.
Okay.
Yes.
Right. So our goal at the working group is trying to support the coordination and access to the genetic variations, and aiming for the agricultural species such as plants, animals, and fish and insect vectors. Our target is trying to enable the sharing of the genetic variation data to support the agricultural practices, and to support the development and adoption for the standards within the community. And so we have to push out some public survey to identify existing genetic variation and try to find out the technical barriers between data exchanges. So we have some engagement activity that could include the survey we have already done on on one webinar, and also coordination with the other working groups to review the workflow for genetic variation and review the technical standards to support genetic variation data within the intent to support the improvements. And we also engage the community to support the ingestion and genetic variation data in archival resources such as EVA. So last month we have an EVA webinar, and here is the QR code. You can scan the QR code to view the videos. So recently, since we have organized the working group, we have a monthly meeting since last November, and we also have a Slack channel for coordination. So if you are interested in what we are doing in this working group, you can join us in the Slack channel to chat, to have ideas about our work. So at the moment, we also trying to recruit additional members, such as Gen+ and curators, to review the standards for Gen+. And also we are encourage some of the non-plans data provider to join us. And we have done one survey during the EVA webinar. So at the moment, we are looking for more broad view. So we are trying to push out the survey by working on IRB approval to support these future surveys. We also have the coordination with other working group. Me as a scientist, I have working with the Pan Genome working group on genetic variation workflow. We also have a review on the EVA data and resources. And since I've said we have an EVA webinar last month, and we do a survey, and later on you can see our summary at the slides. So some of the review are recently published on the recommendation of our variation data. So we are looking to have a recommendation from the VC variation data formatting to make data fair. And recently we have invited Sebastian Bier and Cyril Premier from Elixir Plants to present at AgBioData monthly webinar. So here's an example of me coordinate with the Pan Genome working group for they have a structural variation calling process. And this is a program they call SandPlot to visualize and validate the structural variation. And I have using this program to generate the figure as below. And according to the submission process through the EVA, in February of this year, we have a representative from the EBI, European Variation Archive, that was able to present the monthly AgBio meeting. And EVA has access accept the submission on non-human genetic variation data sets. And Team Cesar, our group members, is a representative from the EVA. And if you have any, if you want to submit or if you want to use the data from the EVA, you can contact with him, with our group working group. So one part of this submission process is linking to other resources at the EBI. One of these is biosamples. And from the recommendations of the formatting on the variant code format, or we call it VCS files. So we are trying to look at the standards to make this plant genotyping data fair. So the Fondue project recently published a manuscript at the F1000 on the recommendation of this plant data. One of the target in our group will be able to review this and implement these for plants and potentially extended these for the other species. So at the moment, we have recruited a plant curator, and we'll be looking for more participants afterwards in breeding and other species. So Sebastian Beer has joined us at the group working group recently. So this is the summary of our survey after EVA webinar. During this presentation, the participants were able to take place in the live poll regarding to how the user preferred to access the data and if they were able to participate in a biocuration workshop. So right now, our challenges at the moment is within the genetic variation is that all the data has a life cycle, and from the old or reused data, so it will generate a lot of complex during the when assessing the variation data, and so migrating data from different assembly or different type of the assembly will cause more complex during when you're using the variation data. And also when you're moving from a single reference to Pan Genome reference, that will causing another conflict or complex. And recently, the improvement of the assay and algorithm to determine the genetic variation will be cause a difficulty to compare between different studies. So we kind of a lack of at the end, we kind of lack of standard for the sample identifier. So it will be hard to integrate between the studies within the biological samples. So that is all the challenges and the work we have done at the moment. So now I would like to have 5 minutes to take a live poll from Anarita.
Yes, I'm gonna launch it. If you guys have any question, this is the time also to ask Rice. Thank you for the very nice presentation, Rice. Someone commented that commented about the polyploid species. Are you guys also thinking about this?
I'm not able to answer. I can't select a response.
I just launched that.
Yeah, I see them, but I
co-host can't take the.
Okay.
I can access them, Marcella. This is Doreen.
Can I have the question again? Kind of lost it.
Yes, they are there. So people, yes, if you want to, you can answer. Yeah, so Rice, are you guys also considering the polyploidy, for what are the difficulties related with polyploidy in crops for genetic variation?
Yes, I think that would be another complex issue when we're looking at the genetic variations. And I think at the moment we are trying to recommend a workflow from all over the different species. So for Diploidy, that would be another questions. That would be another consideration, but it's not. It will be a long term for us to identify it. But I think last meeting, the working group members has mentioned that we should have a short-term and long-term output. So for the short-term, we can just have more simple ideas about the genetic variation and expand it to the more complex issues. I think that would be, that would be more efficient way to set up this kind of standards.
Yeah, so maybe, so I invite everybody that works with polyploid species to join the standard for genetic variation.
Yeah, that would be great.
That great experience can really give a lot of contribution. So people are answering the poll. I will wait for it. If there are any other questions in the audience for Rise.
So the last slide, sorry, the last slide will be our breakout group sessions questions. If you are interested, you can also look at the questions we are going to discuss afterwards. Yeah, and I'm happy to answer the question at the moment.
Yeah, I guess people are busy answering the poll.
Yeah, I do have a question. For example, when a genome assembly is updated, and how do you update, plan on updating the EVA data sets that you have for a particular given reference?
I think I may have to transfer this answer to Tim.
Hi. Yeah, that's a very good question. That's definitely something that EVA wants to tackle and has to some extent tackled already. So all the variants that are submitted to EVA are being updated to the newest version of the reference genome of that particular species. It's a challenge to do that at scale. So it's a good question of whether we will be able to continue to do that for all of the species that we support. But at the moment, the plan is to continue to update them to newer species. So we're pushing them forward most of the time. And there's always a question of what is the most up-to-date or most relevant reference genome for given species. For some species, it's no question. There's only one. But for others, there's often multiple choices, and at the moment, we only want to support one per species. But EVA does plan to update all of the variants that are submitted to us to the newest version of the genome.
Yeah, that's excellent. So one follow-up question about it after that is, as we are seeing more and more of pan genomes coming out, kind of things, right? So as you mentioned that you're using one single reference, but what happens if that particular project or the paper chose to have a different reference genome for their pan genome assembly, and they have the variation data for their project, and now they want to submit to EVA? So would that be a conflict, or how do you envision resolving that? Thanks.
So at the moment, I don't really have a clear answer to that question. The pan genome development is something we're following very closely. So as the community move forward and start to adopt pan genome standard and pan genome infrastructure tooling, we are going to do the same. And so I'm expecting that we will support it as well, but it's not a settled as far as I know, it's not settled yet. What is the right set of tools? What is the right set of standard for representing the genome, and let alone representing the variation on that genome?
Okay, so sorry, Timothy, to interrupt you. I think if you guys want to chat more, you can do it in the chat or during the breakout. So I'm gonna end and you guys, I'm gonna share the results very quickly. And then I'm gonna share the results, download them, and share with you guys of the standard for genetic variations, so you guys can discuss them during the breakout session if you guys are fine. And so with this, I think I want to invite Doreen to share the screen and start presenting about the recruiting working group. Thanks, Doreen.
Do you see my slides?
Yes.
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 Yes, if you can put like full presentation.
Yeah, I did. Hang on a second. I did it wrong.
It's okay, even if it's like that, don't worry.
I still see the standards for genetic, the poll.
We can see your slides. So I just stopped the stop sharing, so don't worry.
Okay, what I did was, I'm gonna do stop sharing.
If you see the poll, just click on the red button on the top left. Close it.
Share screen.
But your slides were there.
To share. Got it. Problem is I have two. Uh-oh. Now you see it?
Yes, we see it. Yeah.
Good. Shoot. Okay. Having two screens, I put it in backwards. Okay, so I'm Doreen Nesdell. I'm going to be talking about the work of the recruiting working group, and my members are my fellow members include Adrienne Brown, Rita Hayford, Lenore Reiser, Anna Rita Morano, Jacqueline Campbell, and Erin Sparks. The mission of the recruiting working group is to ensure equity, diversity, and inclusivity within our working groups and across the consortium. And we will accomplish this by engaging a broadly representative population of stakeholders, including data generators, data consumers, genetics and genomic reading database staff, data curators and managers, data publishers, and funders. Anna Maria summarized the data describing the makeup of the different working groups, from data collection to genome nomenclature to recruiting. She normalized the frequencies for the number of members in a working group so we can compare the working groups. With respect to position, the genotype-phenotype working group is very diverse, since it includes faculty, graduate students, postdocs, research administrators, and other types of researchers. On the other hand, the education working group has a heck of a lot of faculty members. Types of institutions represented was also analyzed for each working group. Again, not all types of institutions are represented within each working group, and the variation in composition is evident. With respect to genome nomenclature, genotype, phenotype, and ontologies, working groups are equally diverse, with members in doctoral, government, independent, and non-US institutions. Genome nomenclature stands out because of the inclusion of master's granting institutions, as does education, with members in both bachelor granting institutions and high school. The overall benefit of joining a working group is that implementing the FAIR principles for data benefits everyone worldwide. FAIR means the data are findable, accessible, interoperable, and reusable. From the individual member's perspective, benefits can include authorship on a white paper, other publications and presentations, networking with colleagues worldwide, and learning new tools and procedures. Our group discussed reasons for what limits a person to join any of the AgBioData's working groups. It may be the focus and the goals are not clear yet. Time commitment may be an issue since we're all busy people, and the skills needed to perform the work may be unclear. As an example, for recruiting is more marketing than understanding genomics. In addition, a person just may not feel qualified or have experienced imposter syndrome. The recruiting working group has been meeting and discussing how we are to move forward. We will be developing a marketing plan that will include developing a list of related organizations and people worldwide and making an effort to target underrepresented groups. We will be developing relationships with like-minded organizations and introduce them to your working groups. We will also develop a social media strategy and presence and add additional details about each AgBioData working group to the web pages. And we are thinking of working with the education working group and developing teaching webinars that include recruitment information. What we would like to know is why you have not joined a working group. And this will be one of the topics of the breakout session. We need to work out how we as members are going to manage the time for all that extra work. And we want to know, is there any way the recruiting working group can help your working group? Since the recruiting working group will be developing a list of related groups, please let us know of any of the groups you want us to include. And for the breakout session, we will be using this matrix to explore how to motivate people to join AgBioData. This example is for the research data librarian, which is what I am. But we have matrices also for other positions, the PI, postdoc, ag researcher in the Global South, breeder at a company, and also journal editor. So thank you, and hope to see you at the breakout session. Any questions? I'm ready to answer them.
Yeah, thank you, Doreen, for presenting about the recruiting working group. So yeah, as Leonor said in the chat, during the breakout session, the recruiting is going to be interested in understanding the barrier to participation and best way to engage. So we really want to know, how do you hear about AgBioData, through which channel? And so this can help us to reach out to more people. Why we need to reach out to more people? Because more people are more experienced, and so we really want to build this community. And so with this, I'm gonna, so if there are no other further questions for Doreen,
I have a question. Can I say it out loud or should I type it?
Yes, go Monica, sorry.
This is great. I was kind of thinking, so there's going to be a breakout group in a bit where some of us will join and discuss with you on these questions, but what you're doing pertains to pretty much all the working groups. So I'm wondering if one outcome or one thing we could do is to have the results of this working group or questions distributed to all of the working groups, just to have everyone consider what their recruiting problems might be after this workshop, because this should pertain to all of us.
Yeah, this is a great idea. I think Leonor, the recruiting working group, prepared a template, and I guess Leonor can share that with in each of the breakout rooms for the 3 days. So we get feedbacks from all of you guys. I think it's very important because, you know, as myself, sometimes we feel discouraged to join any particular working group because we don't think we have the experience for doing that, but that's not true. So are there any other questions for Doreen? If not, so I think it's time that so right now we are going to have a short break, but before that, I want to share with you. So I'm going to open the breakout room, So I just want to, so for whom is not very familiar, so to join any of the breakout room, you see there is this small symbol below your window. So if you click on it, you will have a window appearing, and here there is the list of the breakout room for today. So you can join any of them by clicking the join blue button on the right. So I want to remind the host and the scribe and the guardian that the time for the breakout room is 45 minutes. And I will send an alert that I'm closing the breakout room 1 minute before. It's the time for the host of the breakout room to report about the discussion that you guys had in the breakout room. So I hope everybody's here. So the first working group to report is the genotype-to-phenotype. So I ask Irene to share the screen with slides or anything that you guys want to present.
Oh yeah, I have a, well, let me share my screen very quickly. Okay, so what I have been taking notes here is not very well organized yet. Sorry for the disaster. But yeah, we have been discussing like the following topics. So first of all, we have discussed the current challenges and needs in these phenotype-genotype associations, especially in the context of a multi-omics era. And we have just like come up with a lot of ideas. We have done a kind of brainstorm session. So everybody has shared their thoughts. So yeah, the main concern is how do the data fare, basically, and yeah, in the sense of like in not only in phenotypic data, but also in the genotypic data. So in short, this fairness of the data, as we mentioned. So there have been a lot of ideas regarding that. We have also discussed the minimum standards just to make the data fair, and also how to define a phenotype. So we agree that they, for instance, in the case of gene expression, we can consider it phenotypic data. And we have also discussed a little bit the structure and the scope of the review paper. So what we want to discuss there, what we think that is like are the main challenges nowadays, and yeah, and what things we want to cover in the review paper. So one thing that we have like would like to just state there is that fair doesn't necessarily mean that the data is free. So we all agree that the data has to be accessible to everybody, like open access, and ensure it, and try to figure out the legal aspects regarding this, because not all the data can be accessible because of legal issues. So yeah, this is another key point, make the data accessible. And also take into consideration the evolution of the technologies involved in this data analysis, because they are constantly evolving. So we have come up with the idea of benchmarking, which can be a good option to accomplish that. And also deeply understand the phenotypes. So in order to not only define the ontologies of the phenotypes, but also all the processes that are behind this phenotypic data. So kind of have a more integrative view of the phenotypes, because the same phenotype can be produced, can be the result of different processes. For instance, genes that are like Mendelianly inherited, or a polygenic character, and it's not the same. So we have to also find a way to provide this information to the users. It is huge, but yeah. So basically, we can just like yeah, integrate all the discussion with these main topics.
Okay, thanks, Irene. Okay, so now it's the time of the standard for genetic variation. So Marcela, do you want to share with us your discussion?
Yes, we actually made a few slides, and I want to thank everyone that helped make this. So the first question was right here. What species do you work on or collect genetic variation data for? And we were very efficient in filling out this spreadsheet, and you can see it's mostly plants. But we had one, it was like 19 people in our group, I believe, and we had one representative of animals who works in various species, so that was really great. We are interested in recruiting more animal people because it's a different view. It will really enrich our discussions and our work. Second question, why do you work with genetic variation? We were looking for use cases, right? Like that, it's not just the typical breeder, looking to improve germplasm or researcher, but to do like gather functional information on, or design CRISPR experiments by informaticians who are like working to support stewardship and integrate different types of knowledge. There was also a very specific targeted like domestication or training, like going from a student to postdoc, animal breeding. There was an emphasis on like domestication issue aspects, and also like conservation, looking to preserve species and these rich resources for the future evolutionary analysis and stuff. How do people collect and share variation data? There was a very interesting collection of, because we got several, there are many different formats and different ways, different local repositories or community specific repositories, certain species or certain, clades, taxonomic clades. And then there are other resources that are willing to integrate data from different species. So we really dwell on the need of having standards and a generalized repository, the importance of having one, at least one very broad species for sharing, and the need for the existence of several repositories and several formats, but we need to kind of work together. I think that's the main idea that I got from our session, that we really need to come and agree to something. Otherwise, it's not going to work. We have several different, you can read here, it's very different ways to represent genetic variation data and very different ways to share it with our collaborators. But it would be really in benefit of the community if we could agree on something more standardized and an omni repository. Then we went on the challenges for this particular thing, sharing and reusing, and we have listed here some of the technical ones. The remapping can be like very messy, or the same samples being used in different studies with different sample names. So the need of having a kind of metadata standards, that just the way that we have a variant given an RSID, and there can be several submissions with SSIDs, but they refer to the same RSID, have something similar for samples. And also be aware of, that samples are also going to need to have, geospatial information attached to them, passport information, and all these things. There were also challenges in terms of like policy. Some data can, there is like public-private partnerships. So the owners of the intellectual property may not be willing to share the data because they are, very lines that they have patents or they have worked for, and there could also be, limitations on like public data that is not has not been published yet. There are also the agreements for releasing the data before there's been analysis on it. And we didn't get to actually address these challenges yet, but that's what our group is going to be working on for the next a few months. And then we went to other challenges to integrate now, not only collect and share, but actually integrate with other data types. And I mean, we didn't go on as much detail because actually out of the previous group, what's her name? The previous presenter did a really awesome job in presenting in that one slide, showing the different data types that are integrated, that get integrated nowadays in omic data. So we listed some of the challenges. Funding really was like a big thing, because that limits sometimes not only like the human resources that can be dedicated, not only sometimes even just to curate the data, to format the data. We already know that the standards that we need to work on standards. There are some new recommendations, and we need to like review them and everybody give feedback on them as well. Then we have also other non-data. We didn't touch on much on this, but this is more like referred to like physical access, like to the actual germplasm samples. And the last question was why did anybody like come to this session, and is there anything that was missing? So there were many various reasons: meeting colleagues, connect, experience, training as postdocs, or just learning more about the field, the genetic variation field, gather feedback in the case of like a repository like the EVA, understanding what the community needs, and reducing those barriers to support reusing and sharing of the data. And yes, this is super important. We need to work together to move forward. And we got to recruit our first animal representative in the group, so we're very happy. Mazak agreed, and we're looking for anybody else to join us with this effort, because we really think it's very important. Thank you.
Well, thank you, Marcela. And now it's the last working group, the recruiting. Leonor, do you want to share your screen?
Okay, you got it? Hi. So, hi, I'm Lenore Reiser from the Recruitment Working Group. So we had a lively discussion over a number of different topics, trying to understand what the challenges might be towards participation in working groups. And we, while the sort of filling out the persona matrices didn't really quite work out, we were able to use the example of one of the working groups, the data collection and accuracy working group, sort of as a use case or a test case for what are some of the challenges and barriers towards participation. So one of the biggest challenges, of course, for people is time commitment. So the expectation of the amount of work that's involved. And sometimes that's dependent on the scope of the working groups. So in terms of like how much, what the specific activities of that working group are. So scoping is really important. Some of the other things were communication across different time zones, because we have people from, all across the globe. And we also have this issue that people may not necessarily know that AgBioData exists, or how AgBioData itself can help them with their research, and how participation in AgBioData may benefit them, even if they're not necessarily working on a database. And so sort of thinking about some of the things that are supportive of participation, our people have a genuine curiosity and interest in the topics. People want to be able to educate themselves, and from that curiosity, they find that they want to participate in actually helping to define and build these standards. The other thing would be what's really important is having support from their workplace, from their work environment, to participate in these sort of extracurricular activities. And then having sort of more available resources at AgBioData to help inform people about what AgBioData is doing and how that might benefit them. So some of the things that we can be working with the various working groups on are making clear in the scope and the deliverables, working on the modes of communication, and then within the recruitment working group is identifying and reaching out to more individuals and potential stakeholders to spread the knowledge and information about AgBioData and engage their participation in the consortium and in the working groups. And I will stop there.
Well, thank you so much for all the people that joined the discussion today, and you guys did a really great job. So with this, I want to share the screen and conclude this first day of the community workshop, which is really great. So I want just to remind the working group that the feedback that you received today will guide your work for the following month, and that we want to hear more about how you are going to implement the discussion of today in your work during the next AgBioData webinar on April 6th. And with this, I want to invite you to join us tomorrow. We will have the 3 working groups. We will have the education, the genome assembly and annotation nomenclature, and the Pan Genome working group. So thank you so much for joining us today, and I hope to see you again tomorrow. Thank you.